Bun găsit la emisiunea Știință și Cunoaștere. Revenim la biologie. În perioada ultimelor zile ale anului 2013 lecturam pe internet câteva noutăți legate de transportul inter- și intracelular, fiind deja pregătite 3 emisiuni cu profesorul Octavian Popescu, în care am aflat noutăți excepționale în biologie. În cadrul uneia dintre episoade, domnul profesor a amintit faptul că această descoperire aparține unui colectiv format din 3 cercetători cărora li s-a acordat Premiul Nobel pentru medicină sau fiziologie în luna noiembrie 2013. În timp ce lecturam toate detaliile acestei descoperiri pe paginile de pe internet, la un moment dat am găsit o adresă de e-mail a uneia dintre cei cercetători, și anume profesorul Randy Scheckman, care avea experiență în științe biomedicale. Fără niciun fel de speranță, am conceput o invitație pentru a realiza un interviu prin camera web. Nici nu mi-a venit să cred când a doua zi dimineața, în 29 decembrie, am primit răspunsul că acceptă să facem interviul. Începând din acel moment am pregătit structura emisiunii și la un moment dat l-am sunat pe telefonul de la birou, dar nu era acolo, însă vocea robotului automat mi-a indicat un alt număr pe care la început nu am fost în stare să-l notez la prima audiție, așa că am sunat a doua oară ca să completez cifrele și am reușit să ne auzim la început de ianuarie 2014, stabilind o întâlnire de probă pentru a regla sunetul și luminile. La începutul lunii februarie am înregistrat aproximativ 48 de minute. Acesta este primul interviu cu un laureat al Premiului Nobel din cariera mea și probabil printre foarte puținele interviuri realizate în România de alți jurnaliști. Este cea mai mare onoare de care se poate bucura un jurnalist de știință. De mic copil, Randy a fost atras de cunoașterea bazată pe experiment și rațiune. În perioada anilor de dezvoltare profesională la Universitatea Stanford, Randy se va căsători cu Nancy Walls și va avea 2 copii. Își continuă munca în cadrul mai multor laboratoare alături de Arthur Kornberg. În prezent, Randy Wayne Scheckman este cercetător în domeniul biologiei celulare la Universitatea California, Berkeley și membru al Academiei Naționale de Științe. În anul 2002, Randy Scheckman a primit împreună cu James Rothman premiile Louisa Gross Horvitz și Albert Lasker pentru descoperirea mecanismului transportului membranar. Celor doi li s-a alăturat mai târziu și Thomas Sudhoff, care toți 3 împreună au câștigat Premiul Nobel pentru fiziologie sau medicină în luna octombrie 2013 pentru descoperirea mecanismelor care reglează transportul veziculelor, un important sistem de transport în celulele noastre. Randy este un om deschis, sufletește și comunicativ, deosebit de modest și este în permanență fascinat de noile descoperiri. Profesorul Randy Scheckman are rude în Basarabia și conexiuni aproape sufletești cu România, deoarece în munca sa neobosită a colaborat cu academicianul George Emil Palade și pe care îl consideră mentorul său, menționând în cadrul interviului noutăți și recunoașteri niciodată prezentate până acum la vreo televiziune la noi în țară privind contribuția României la patrimoniul științific medical internațional datorată academicianului George Emil Palade. Mi-a povestit cu emoție despre modul în care academicianul George Emil Palade și-a prezentat propria descoperire în domeniul biologiei celulare în perioada anilor 70, iar la final mulțimea din sala de conferințe l-a aplaudat mai multe minute, ridicându-se toți în picioare, cuprinși de o stare de revelație fără precedent. Un asemenea nivel de apreciere reprezintă un fapt ce nu a mai fost consemnat până acum. Pe drept cuvânt, profesorul Randy îl consideră pe Palade ca fiind un deschizător de drum și un cercetător genial. Randy continuă să predea studenților materiile de biologie celulară. A donat întreaga sumă de bani pe care a câștigat-o cu ocazia premierei sale la un institut de cercetare în domeniul cancerului, deoarece mama și sora lui Randy au murit de cancer. Vă invit acum să vizionați prima parte a interviului.
Hello, my name is Professor Randy Scheckman from the University of California, Berkeley, and I won the Nobel Prize for Medicine or Physiology in 2013. And you're watching Science and Knowledge on TVR Cluj, Romania.
Dear Professor Randy Scheckman, welcome to our Science and Knowledge program on TVR Cluj for the first time in Romania.
Aha, great. Good to be with you.
For me, as a journalist, it is the greatest honor to have you as guest in this program, which is based upon public understanding of science, and I have not enough words in my poor English to express the gratitude and my deepest thanks for accepting this interview.
My pleasure, my pleasure. As I think you know, my ancestors are from Romania, so I'm pleased to say hello to anyone who from my family who may still be there.
Yes, and I am happy that you have connections with us, and if I remember correctly, in Bessarabia?
That's right, that's right. Mm-hmm.
In Bessarabia, okay. Also, due to the importance of this interview, I would like to consider that there is no specific limit of time. Eventually, we may prolong the duration in order to record the complete answers for me and our viewers.
Sure, happy to help.
On the introductory part, please present us a brief history of where your research started related to this amazing discovery of mini vesicles transport, and please explain the mechanism for the untrained and non-specialized public.
Sure, sure, I'm happy to do that. So most of the cells in our body make the molecules called proteins that are the catalysts of life. Proteins, for the most part, do all the chemistry that makes a living cell unique. Humans have around 23,000 genes. That means at least 23,000 different proteins. And about 10% of these proteins, on average, are designed to be shipped outside of a cell. That is, all proteins are made inside of a cell, but some of them are special proteins that have to be exported from the cell, and they go into, for instance, the bloodstream, and they attach themselves to other tissues. For example, insulin is made in the pancreas, and it's exported by a process called secretion. And it goes into the bloodstream, and it circulates around, and then it attaches itself to target cells that respond to sugar when you have a meal, and allow the sugar to be taken into cells and to be stored for energy. Other proteins are in your blood, antibody molecules that fight infection, lipoproteins like LDL and HDL that we of a certain age know about. These are manufactured in the liver, and then they have to be shipped outside of the cell. So all cells, all growing cells, from bacteria to man, have this capability of manufacturing some protein molecules that have to be shipped outside of the cell. And that presents a special problem because the cell wants to stay intact and to hold on to most of the proteins that are operating to do the chemistry of life. So how does it let some proteins, some unique proteins like insulin, selectively escape? That's the challenge. So over many decades, beginning in the early 20th century, physiologists wanted to understand how this could happen, how proteins could be secreted. And there were many classic studies on this that speculated on the existence of an intracellular machinery, a kind of a conveyor belt-like machinery that may be involved in encapsulating proteins that are going to be shipped outside of the cell. And the crucial experiments that led to our understanding of this in the latter half of the 20th century came from one of the great Romanian scientists of history, a man named George Palade, who did his pioneering work in New York after he moved from Romania. Palade, among other things, discovered a very special machine called the ribosome, which is responsible for making proteins. It does so by stitching amino acids one after another to make a long chain, which then folds into a compact shape to give each protein molecule its unique function. He discovered the ribosome. He also discovered, using the technique of electron microscopy, how the membranes inside of a cell are organized with respect to each other, eventually to create these capsules, which are called vesicles, that surround the proteins in a container and then ship them to the cell perimeter, where they are discharged to the cell exterior by a process called fusion, where the membrane that surrounds the container literally merges with the surface of the cell by fusion, and that causes the internal content of the container to be spilled outside of the cell. So that's a unique process, and Palade was the real pioneer in the 1960s. He discovered the orderly events that execute this process. He studied them largely in the pancreas, in the mammalian pancreas.
During your career, did you meet in person George-Emile Palade?
Oh yes, very, very, very many times. Yes, I first met him when I was a graduate student at Stanford. He was a good friend of my graduate mentor, a man by the name of Arthur Kornberg. And Kornberg thought very highly of him and was, I gather, instrumental in Palade winning the Nobel Prize in 1974. So I heard Palade lecture on his work as a graduate student, and then I remember vividly when I was, after my PhD, I did a postdoctoral period of study in San Diego, California, and Palade had just returned from Stockholm after having been awarded the prize. And he gave a special lecture at a big national meeting in the US. And I'll never forget the description of his experiments. And he received a standing ovation by the audience at the end of this lecture. I had never seen that before in a scientific lecture. So he was revered as a really profound scholar of this area. And I got to know him more and more as the years went on, because I then joined that field. I had not worked in that subject area until I was after my PhD. And so he and I met on many occasions, and I had the opportunity to speak at symposia in his honor, both when he was alive and a special symposium in his honor after he died. And I wrote an appreciation piece, an obituary on him that was published in Science magazine shortly after he died. So I had the utmost admiration for him. He was really a brilliant scholar and very broadly knowledgeable. Anyway, so but what he discovered was, how this process was organized. But what wasn't possible in his work was to look at, with greater precision, at the molecules that actually execute this pathway. So the cell organizes this pathway, and it must use special machines, special proteins, to allow this regular event to be organized. And the techniques that he used were not of sufficiently high resolution to understand the mechanism. So what we want to understand is the mechanism, how it actually is that these little capsules are created, and how they find their way to the right target, and how they fuse. So we want to know this at a molecular, even an atomic level of resolution. So how do you take that step? So what I did when I started my career here at the University of California, Berkeley, was to take a step back and to not use mice or other mammalian sources, as had been done by Palade and others, but rather to imagine that the same process may have been evolutionarily conserved for so long that even something as simple as baker's yeast might have the same machinery. And indeed, it was known that baker's yeast, that we all know in the cooking and brewing industry, is in a way like a mammalian cell in the way that it's organized. It has a nucleus where the chromosomes are contained, and it has membranes inside the cell that look a little bit like what you see when you cut a thin slice through a mammalian pancreas.
And is that a reason for you to decide to use yeast in order to identify the key genes for vesicular trafficking?
Yes, of course. It turns out that baker's yeast is not only simple, but it's technically very easy, much easier to use molecular techniques like genetics or biochemistry to understand a fundamental process. So we began in 1976 to apply genetic techniques to identify the genes that control the pathway that Palade had revealed. And we were able to discover many genes that, when interrupted by mutation, block this regular pathway at very specific stages. And over time, having been able then to clone these genes, we can understand what the proteins that were produced by these genes did, how they catalyzed these events. And so we went from a genetic analysis to a more biochemical analysis, where we were able to isolate molecules and see how they interact with one another in the test tube.
What is the importance of these genes for life itself?
Well, these genes are essential. If you cripple these genes, even in lowly yeast, the yeast cell dies. And the same is true in humans. There are some diseases where these genes are affected, and they can be very severe. And even so, these are only weak mutations. A strong mutation in these genes would surely kill a human being. But weak mutations have very specific pathologies that can be recognized. So there is a commonality, and this is what motivated our search in the first place. There's a relationship over 1 billion years of evolution that has conserved this machinery. It was invented over 1 billion years ago, and it operated in an important fashion to allow the cell to grow and nourish itself. And so the bits and pieces of those machines have been used over and over again throughout evolution. And even now in the human being, these same proteins that we discovered in yeast operate in the brain, they operate in the liver and the pancreas, all over the body.
Are the glycosidase and the glycosyl transferase genes involved too in the vesicle genesis and their transport to target site?
Yeah. So many of the proteins that are exported from the cells have sugar molecules on them that are manufactured by enzymes that are in the membranes. Glycosidases and glycosyl transferases that you mentioned are in the membranes that organize the traffic, and they decorate proteins in specific ways. And some of them are important to allow the protein that are about to be exported to fold into the right conformation. And so when you have mutations in some of these genes, you make proteins that don't fold properly, and they get degraded. They get broken back down into amino acids. Sometimes the sugars that are attached allow a protein to be shipped to a particular destination within a cell. And instead of being secreted, they may be diverted to an internal organelle that's involved in protein degradation, of an organelle called the lysosome. So the glycosylation reactions are also important.
How could your experimental data can be extrapolated to humans?
And I can give you two examples. As I already mentioned, there are some diseases, some genetic diseases, where the genes that we discovered in yeast are mutated in patients. And we've been studying some of these diseases. They're fairly rare, but they're quite severe. And we can understand the nature of these diseases because we have already developed tools to study these processes in yeast, and the same tools can be used to study these processes in mammalian human cells grown in a culture medium in the laboratory. So there is a direct connection through human disease. Another connection is when we realized in our experiments that yeast cells use a process that's fundamentally the same as in human cells. The biotech industry, which grew up here in the San Francisco area, was able to harness yeast cells as a kind of a factory to produce useful quantities of human proteins. And the best example would be human recombinant insulin. So if you have diabetes, now you don't have to rely on the insulin that was produced from pig pancreas for many years. Instead, you can rely on a recombinant, authentic human insulin. And 1/3 of the world's supply, indeed likely most of the supply in Europe, is made by secretion in yeast. Because a local company discovered that if you take the gene for human insulin and you put that into a yeast cell, you can cause that gene to be recognized. The yeast cell can be fooled into recognizing it as one of its own genes, and it will copy the gene and make the human insulin protein. And since this pathway of secretion is the same, the human insulin, once it's manufactured inside of a yeast cell, will hop onto this pathway and be secreted outside of the cell. And so you can use a fermenter to grow up thousands and thousands of liters of insulin in the growth medium. So that's another example of the intimate connection between the human process and the process in baker's yeast.
This is really amazing, and I'm sure they will have a lot of consequences on the development of new medicine, and I believe mostly on pharmacogenomics, because you can send the specific medical drug to a very specific target cell using physical transportation and minimum side effects.
Yeah. So again, learning the basic mechanics from yeast allows one to discover the equivalent mechanics in the human cell. And so it might not be so obvious to your viewers that it's sometimes simpler, easier, more direct to study a problem not in humans, but in an organism that is simpler to grow and manipulate in the laboratory. And yet the lessons that you can learn from something as simple as baker's yeast can actually apply quite directly to what happens in a human cell.
Why did you categorize these genes in 3 classes?
Okay, well that was on the basis of the appearance of these cells in the electron microscope. What kinds of organelles are blocked by the distinct classes of mutations. The first organelle that we found that was blocked in the first mutation are mature secretory vesicles at the end of the pathway. And then later, when we found more genes, we found 2 other classes. One where traffic from this structure called the endoplasmic reticulum is blocked, and therefore that organelle builds up in the absence of movement to the next stage in the pathway. And the third class was a mutation that causes the structure called the Golgi apparatus to build up to a much higher extent than is normally seen in a yeast cell. And so that was those are the 3 classes of organelles that we found in our mutant cells.
În partea a doua a interviului, profesorul Randy Scheckman va vorbi despre corupția existentă la nivelul celor mai importante reviste de știință ale lumii. Pe lângă ceea ce veți afla direct de la domnul profesor, îmi permit să mai citez câteva lucruri pe care domnia sa le-a publicat la începutul lunii ianuarie și veți avea afișată pe ecran adresa de internet unde puteți afla chiar și mai mult de atât. Sunt un om de știință, lumea în care profesez realizează lucruri mărețe pentru omenire, dar este desfigurată de stimulente neadecvate. Structurile predominante reprezentate de reputația personală și de avansarea în carieră sunt cele mai mari recompense, dar de multe ori acestea vin în urma muncii care nu este neapărat cea mai bună, ci a celei care iese cel mai mult în evidență. Aceia dintre noi care urmează aceste stimulente sunt complet raționali, dar nu reușim să servim întotdeauna cele mai bune interese ale profesiei noastre, cu atât mai puțin ale umanității și ale societății. Știm cu toții ce au făcut stimulentele denaturate în domeniul financiar-bancar. Stimulentele în fața cărora sunt puși colegii mei nu sunt bonusuri imense, ci recompense profesionale ce acompaniază publicarea în reviste de prestigiu precum Nature, Cell și Science. Aceste reviste de lux ar trebui să fie simbolul calității și să publice cele mai bune lucrări, deoarece comisiile de finanțare și numire folosesc revistele ca pe un argument al calității științei, publicarea de articole conduce de multe ori la obținerea de burse și catedre. Deși revistele publică lucrări excepționale, ele nu publică doar lucrări excepționale. Aceste reviste își administrează agresiv brandul în moduri care mai degrabă favorizează vânzarea de abonamente decât stimularea celor mai importante cercetări. La fel ca în cazul designerilor de modă care creează genți sau costume în ediție limitată, ei știu că deficitul alimentează cererea, astfel că restricționează în mod artificial numărul de lucrări pe care le acceptă. Brandurile exclusive sunt apoi comercializate prin intermediul unui truc numit factor de impact. Un scor pentru fiecare revistă care măsoară numărul de citări ce apar în cercetările ulterioare. Teoria spune că cu cât sunt mai bune lucrările, cu atât sunt citate mai des, deci revistele mai bune se pot lăuda cu scoruri mai mari. Cu toate acestea, aceasta este o măsură profund viciată și este la fel de dăunătoare pentru știință, așa cum este cultura bonusului pentru domeniul bancar. Este o practică comună și încurajată de multe reviste că cercetarea să fie judecată după factorul de impact al revistei în care este publicată. Dar în cazul unei reviste cu un scor mediocru, aceasta spune foarte puțin despre calitatea fiecărei cercetări în parte. Citarea este câteodată, dar nu întotdeauna, asociată cu calitatea. O lucrare poate fi foarte citată pentru că este foarte bună din punct de vedere științific sau pentru că este atrăgătoare, provocatoare sau chiar greșită. Editorii revisturilor de lux știu asta. Așa că acceptă lucrări care fac valuri deoarece ele abordează subiecte sau fac afirmații provocatoare. Am încheiat citatul și vă invit în cele ce urmează să vizionați partea a doua a interviului. Now, on the second part of our interview, it would be important to emphasize something about how top journals like Nature, Cell, and Science are damaging the science and the scientific discoveries. Please also explain for our viewers what is a peer review and how it is done.
Yeah. Well, so scholars around the world do their work, and it's crucial that they publish their work so that others can see their results and can repeat their results. It really, a scientific discovery doesn't mean anything until someone else can repeat the results and show that they're valid. And so we publish. And there are thousands of journals all over the world. So there are many opportunities to publish work. But scholars, of course, want to have their work in the most visible journals so that their colleagues can read and see and take note that this work has been published. And over the years, several journals have risen to the top. They're usually commercial journals. And these are journals, particularly Nature in Britain, Science, published in the US. And then in life sciences, a journal called Cell, which is run by Elsevier Press, a European major publisher. These are, in the case of Cell and Nature, commercial, for-profit entities. Science is run at a profit to support the activities of the American Association for the Advancement of Science, which is a non-profit organization, but the journal is there to generate profit for that organization. So these journals have very successfully encouraged scholars to seek those very few journals as a venue. And as a consequence, all young scholars who seek to advance their careers feel desperate that they have to have their work published in these very few venues. And this creates tremendous pressure. These journals are, in a way, a victim of their own success. Tremendous pressure to find space in journals that still rely, to a large extent, on a print copy. So Science publishes 50 pages a week, Nature something like that. And this it has to cover all of science. So it's very difficult because of this restriction, which I consider in the 21st century to be an artificial restriction, because most journals now are online. All journals are online, and most young scholars read the literature online. They don't read the print version. And yet, Science and Nature in particular base their decisions about what gets accepted on the print version. And that is, I think that has created a distortion. And so the distortion is that in the case of Science, only something like 6% of the papers that are submitted get accepted. In Nature, it's 8%. That means that people are desperate to get their work published, and sometimes, unfortunately, they may cut corners to make the work look more interesting, more appealing than it actually is. So very often, work published in these journals turns out not to be reproducible. Sometimes the papers have to be retracted. And these journals have a commercial interest in creating this false commodity. I call these journals luxury journals, and I've compared them to the ladies' fashion industry, where haute couture designers limit the number of copies of exotic articles of clothing and to be able to increase the price, to make it seem of a more precious commodity. And yet, in this case, we're talking about publications that could be online and for which no space restrictions need apply. And yet, these journals restrict themselves in this way and make it very difficult on scholars to publish their best work in these venues. So I've criticized the business plan of these journals, and I've asked for my colleagues to consider publishing their work in open access journals where anyone around the world can read the paper without having to have a special license to do so. For example, people in your audience may wish to read about the detailed discovery of something that they learn about in the newspaper, and unless they're at an institution that subscribes to these scholarly journals, they don't have access unless they're willing to pay a fee for just the privilege of reading a publication. Sometimes it can be the equivalent of $30 just to read a paper. And these are papers, by the way, these are scholarly works that have been supported by an investment of various governments to do the science. So it's a public investment in the execution of the work, and yet you as a citizen may not be able to access these publications freely. In spite of having paid for them, you may not have access to them.
Not to mention that sometimes bad science had been published in Nature and Science.
Yes, of course, there's always that, and I think that's part of the problem, that there's a tremendous pressure. And one example that I give is that in mainland China, the major agency that funds science in China, called the Chinese Academy of Sciences, has offered their scholars what amounts to a bribe of the equivalent of $33,000 merely for the success in publishing a paper in the journals Science, Nature, or Cell. And this is personal spending money. It's not money for their laboratory. So it's basically a bribe to get their scholars to be successful in these journals that amount to a lottery system. And so imagine the pressure that a young scholar faces for advancement, for personal income, to try to push to get their work published in these journals. I fear that this leads scholars to cut corners and maybe to misrepresent their work. Now, of course, these journals publish great papers. Naturally, they understand what constitutes good science. But I think very often they're looking for papers that create a lot of buzz, that will generate a lot of attention, papers that will be cited by others. So they rely on large numbers of citations to sell themselves in the marketplace through something called an impact factor, which is a kind of a business gimmick that journals use to advertise themselves. It's just basically the number of citations that their papers achieve over a period of 2 years. This is, I believe, a false measure of scholarly achievement. And yet journals, particularly Nature and Cell, rely on the use of this impact factor in their business plan to sell magazines. And this is a distortion, and I've called on my colleagues to resist the temptation and to publish instead in open access journals.
And related to what you had just said, it is important to say that not long ago such an open access journal invited me to write a paper. Even I am not a biologist, but I have a personal experience which I could explain in scientific terms, which are still in the public understanding of science area, not fundamental science. And they published it, and it was for free.
Well, there are many such open access journals now. Some of them are more reputable than others. Unfortunately, some of them are simply there to make money also by charging the authors page charges for the publication of their work. So you have to be wary of this.
I can give you an example about bad science being published in Nature magazine, and I'll explain for our viewers the problem while the text is uploading to you. and they are from Nature Volume 382, July 1996, where the so-called damage appeared, and the second text is from Nature Volume 384, December 1996, on the errata. On the first volume, they published a wrong article about climate changes, which was probably sponsored by some agency or governmental institution who had enough money to pay, and it was proved wrong. And then some professors from the Czechoslovakia criticized them, and they had to publish for the prestige of the journal. And I have many examples like this. And because I have some time to insert this, I'll present a second example in Romanian language, but with English subtitles in order for you to understand my later on inserted and edited presentation. Ca și o curiozitate, sigur, mult mai târziu, Nature Magazine a publicat în anul 2011 un articol care discreditează în totalitate orice influență a omului asupra climei și găsim următoarele afirmații. Revista de știință Nature, clasificată ca fiind cea mai bine cotată publicație științifică periodică din lume, a publicat un studiu despre încălzirea globală în care se dovedește faptul că forțele dominante care controlează temperatura atmosferei Pământului se datorează radiațiilor cosmice și a Soarelui și nu a omului. Unul dintre autorii acestui raport, profesorul Jirki Haupinen, a rezumat concluziile sale cu privire la factorul antropic asupra încălzirii globale, spunând că, citez, eu cred că este o falsificare strigătoare la cer. Am încheiat citatul. Avem și aici adresa originală pe care oricine o poate accesa și aș mai explica de asemenea telespectatorilor, în cazuri precum acesta nu e nevoie să fiți experți în climă pentru a înțelege ce se vede aici pe acest grafic. Aici avem, de exemplu, variația temperaturii versus concentrația de CO2, dioxid de carbon, pe o perioadă de timp extinsă. Și acesta este un grafic oficial al UNEP despre schimbări climatice în domeniul public, care este prezentat gratuit pentru informare științifică. Iată acum exact același grafic, exact aceeași reprezentare, dar la care s-au adăugat câteva linii verticale paralele pentru a observa că, în realitate, nu creșterea concentrației de CO2 determină creșterea temperaturii, ci invers, adică imediat după ce crește temperatura, crește și nivelul de CO2. Dar mai important de atât, atunci când temperatura scade, se vede că și nivelurile de CO2 scad. So this supplemental comment is for our viewers. But coming back to our first example, can we imagine something like this?
Yeah. Well, I don't have time to read this right now during our conversation.
But you can keep them.
Yes, I have. I'm looking at one of them right now in Nature. Well, there's a lot of in the area of climate change. There's a lot of very heated discussion. I mean, most scientists who are active in the field understand that there is climate change and understand that there's a human role in this process, but there are some scientists who dispute that, and there continues to be a lot of political discussion on this.
Yeah.
Anyway, so I am the editor of one of the new open access journals. It's called eLife. It's the eLife journal is sponsored by three of the very most influential science organizations in the world: the Howard Hughes Medical Institute, the Wellcome Trust, and the Max Planck Society. And these organizations felt strongly that the system was broken, that their investigators were not able to publish their best work in a timely manner, and they felt that there was a better model, not just open access, but where the literature is actually read and decisions are made by active scientists. And so I took on this task starting a couple of years ago, and we've now published this journal for over a year, and we're very actively collecting important papers that we wish to publish online. Well, I'm just in promoting this new journal, eLife, I wrote an article, an editorial for The Guardian that was published just on the day that I won the Nobel Prize in December. And this created a lot of discussion, much more discussion than I would have guessed. And it continues to occupy a lot of my interview time. People are very interested in either supporting or opposing my views on these so-called luxury journals. But these journals, of course, are very powerful. They will continue. They have big financial backing, and so it's a hard sell to convince young scholars to consider a new journal to publish their most important work. So we're working very hard to convince people that we have a better way of reviewing and publishing their work, and so this occupies a lot of my time.
And I'd like very much to offer you all my support for your journal and your activity. And if you agree to keep contact with you from time to time, I'll be delighted to do so.
Great. Happy to have your help.
În ultimele aproximativ 10 minute care au mai rămas disponibile, mi-am asumat riscul de a îi prezenta domnului profesor Randy o ipoteză medicală care pornește de la ideea transportului vezicular inter- și intracelular și se referă la posibilitatea utilizării mitocondriilor pentru stimularea fenomenelor de regenerare tisulară în cazul maladiilor degenerative. A fost surprins de preocupările mele în domeniul medicinei mitocondriale, care este o nouă ramură a medicinei ce se dezvoltă momentan într-un singur institut din lume, dar a apreciat ipoteza mea ca fiind plauzibilă și realizabilă. Fapt ce mi-a deschis noi direcții de orientare a anumitor preocupări în domeniul cercetării de avangardă, dar și al jurnalismului de știință, deoarece intenționez să scriu o carte despre medicina mitocondrială, care schimbă sau urmează să schimbe întreaga paradigmă medicală legată de bolile degenerative, oferindu-ne o nouă viziune. Dear Professor Randy Scheckman, for the next part of our program, I'd like to discuss with you a hypothetical situation based upon the abstract of some important peer-review published papers, if you agree.
Okay.
And where I'm wrong, please stop me and explain the correct thing. But again, I emphasize the fact that this is nothing more than a logical discussion, but your word count a lot for me and our viewers.
Okay.
I must take into a direct example where your discovery might be involved. Everybody here or know what is a lumbar discopathy or many other intervertebral problems. Let's see what was the old medical paradigm for that. Medical science said, sometimes a disc swells, tears, or degenerates without any apparent cause. An intervertebral disc may go into a degenerative process either by deficiency in collagen or in fluid cores or even water deficiency. And here is the original abstract of that paper.
Mm-hmm.
And please do not close the Skype windows instantly after our talks in order to save all those links and papers that I am prepared to send you and talk about.
Okay.
And now here is the new paradigm. This is very interesting. According to the next article published in Cellular and Molecular Bioengineering in June 2011, Volume 4, Issue 2, pages 302 to 310, degeneration is the inability to maintain extracellular matrix integrity. Extracellular matrix synthesis is closely related to production of adenosine triphosphate, EA energy of the cells, quote closed. And here is the original abstract of that paper.
Okay.
But the most important piece of the cell responsible for the production of ATP in the cell is the mitochondria.
Yes. Mm-hmm. Of course.
And while some cells do have 100 or almost 1,000 mitochondria inside, like our muscle cells, other cells have only 1 or 2. And now here comes a new discovery, which said, and I quote, "It has been reported that human mesenchymal stem cells can transfer mitochondria to the cells with severely compromised mitochondrial function. Isn't it amazing? They can transfer mitochondria. And here is the original paper.
Yes. I've seen actually there was a paper submitted to my journal on that phenomenon of transfer of mitochondria from one cell to another.
Using vesicles. And let me back up even more my assumption, explaining based upon another published paper that there are means of transportation of mitochondria within vesicles, which is your discovery, and replace the malfunctioned one. Experiments done on mice revealed that this is the case, and I quote, "A new study using a mouse model of lung disease is the first demonstration in vivo that bone marrow-derived stromal cell can repair tissue injury through the transfer of mitochondria, and this suggests that rescue of injured cells through mitochondrial transfer may be an important process in many diseases. Much like the initial response of classical physicists to the discovery of subatomic particles, most biologists are skeptical about cell-to-cell communication that occurs through vesicles or particles. It's perhaps too messy and confusing. Nevertheless, there is a conclusive evidence that cells can communicate through the transfer of vesicles and particles carrying lipids, proteins, messenger RNA, microRNA, and DNA. Quote closed.
Yes.
And here you have the original paper.
Actually, this is not.
I'm coming to something, if I may. I am following a very logical construction here. So if you can stay with me for 1 or 2 minutes more. It is also known that in ancient times, mitochondria existed as prokaryote cells, and later they formed together larger eukaryote cells. The division between prokaryotes and eukaryotes is usually considered the most important distinction or difference among organisms. And amazingly, let me also explain what this abstract of a new paper entitled Mitochondrial Transfer Between Cells Can Rescue Aerobic Respiration. And this new paper is talking about, and I quote, "Recent reports have found that mitochondria play essential roles in aging and determining lifespan. A variety of heritable and acquired disease are linked to mitochondrial dysfunction. We report here that mitochondria are more dynamic than previously considered. Mitochondria, or mitochondrial DNA, can move between cells." Quote closed. And now I am coming to the idea which is completely new, and I am hypothetically saying that degenerative diseases like lumbar discopathies can be cured using mitochondria transfer from a special group of cells which can play the role of donor cells in order to restore the extracellular matrix of the intervertebral disc. And I have done this on myself, and I have the MRI results before and after this unusual event of biological transformation, and also other analyses which can indicate that not only I stopped the degenerative process of a 15-year-old lumbar discopathy, but even more than that, I started a regenerative process to its earlier stage of the disease, which cannot be clinically detected, but only using advanced medical devices. So I have good reasons after studying your discovery that this healing had been performed using mitochondrial transport in the vesicles, your discovery. And I was later on becoming involved in a collaboration with a professor from Houston Victoria University, a campus of the Houston University's system in Texas. And I wrote this book where I had explained the method of how I did this healing, but only recently I was thinking more in depth to this amazing mitochondrial transportation mechanism. And if my logical approach is correct, then I'd like to continue to be in touch with you for advice, because finally, if anything might come up from this idea, it could be useful for other patients.
Yeah, okay. Well, I didn't realize that you had such a direct interest in actually doing science yourself, but this process is plausible. Whether it can be applied in therapy would, of course, require clinical trials.
Yes.
Very often these sorts of things start off looking interesting and promising, but in the cold reality of clinical trials, they unfortunately usually fail. And there's a lot, for instance, a lot of excitement about using stem cells or using adult stem cells in therapy. And unfortunately, without a proper clinical trial, people get their hopes up about therapies that prove not to be realistic. So, but transferring mitochondria between cells is certainly plausible. It may be important in certain contexts. And it may be even useful, but until there's actually a serious clinical trial, double-blind clinical trial, one must remain skeptical.
I totally agree with you. I am 100% agree with you, and this is why I formulated all this only as a medical hypothesis and nothing more. But can you tell me what kind of markers should be used in order to visualize that mitochondria can travel in the vesicles from a specific donor cell to a target damaged cell which needs a mitochondria replacement? What kind of marker should be used and how?
Yeah, that's an easy thing to do. What you do is you tag a protein that's in the mitochondrion with a fluorescent protein. There's a protein called green fluorescent protein, and if you fuse the gene that encodes green fluorescent protein to the gene that encodes the mitochondrial protein in a donor cell, you will make the mitochondria in that donor cell fluorescent, so that you can, in real time, in living cells, visualize the fate of those mitochondria. If you then mix those cells with a target cell that you believe may acquire mitochondria from the donor cell, you will be able to visualize that in real time by the appearance of the fluorescent mitochondria from the donor cell in the acceptor tissue. So that's a very feasible way of doing that.
Is that something similar to what had been also awarded with a Nobel Prize in chemistry a few years ago? And I found this paper here. Is that the technique that we need?
Yeah, so this protein, green fluorescent protein, was the subject of the Nobel Prize in Chemistry several years ago to three U.S. scientists. Roger Chen was the lead investigator who applied green fluorescent protein in various biological systems, and this is the technique that I'm referring you to. Well, what I've described is a fairly routine laboratory technique, but of course it requires a laboratory to do it. You can't do it in your kitchen, so you'd have to, if you were interested, you'd have to have a collaborator who was able to do this with you.
Yes, the very unfortunate situation for me is that I am not an academic. But on the other hand, I am among those very few who could recover from a pretty advanced lumbar discopathy of 15 years of pain.
Oh my.
And I needed first to discover how to do it. And it took me all these years to learn how to do it.
Okay, well, I have to caution you, I'm not a physician. I work with cells, I don't work with, whole organisms, so I'm not really the right person to consult here, except I know about cells, and the basic question of whether mitochondria can move from one cell to the other is something that could be addressed in the laboratory.
Yeah. So until now, you believe this do have a logic?
Yes, this is plausible.
Dear Professor Randy Scheckman, it had been an amazing privilege and fascination for me to talk with you, and I'm so happy that we did this first interview, and I hope that you agree to do it once per year, every year, if you agree.
Okay, well, you can get back to me and we can talk about other interesting issues in biomedical science, sure.
And if I'll need some ideas for this plausible and possible future research, may I have your permission to address to you for an advice?
Sure, so we can correspond by email.
As soon as the program will be aired on TVR Cluj, the entire recording will be publicly available on a YouTube link, and you may share it with your colleagues and friends.
Okay, thank you, Christian.
I thank you so much for your explanations, considerations, and excellent presentations. And it was a real pleasure to have this conversation. And also for me, in the last part, this had been one of the most difficult exams.
All right. Well, I hope your ideas prove fruitful.
Me too. I hope also, and I thank you a lot.
Okay. All right.
Thank you very much.
Have a good weekend.
You too.
Bye, Christian.
All the best. Bye.
Bye.
Aceste 10 minute au însemnat pentru mine un examen destul de dificil și nu știam dacă voi fi în stare să-l susțin cu argumente suficient de serioase pentru a fi luate în considerare. Discuția a durat mai mult de 10 minute, dar am selectat o parte din ea, iar conținutul integral al ipotezei prezentate îl veți putea lectura ulterior pe internet. Trebuie să mai adaug faptul că întâlnirea cu profesorul Randy Scheckman a fost una dintre cele mai emoționante, tulburătoare și fascinante din câte am avut până acum la Știință și Cunoaștere. Doresc să aduc sincere mulțumiri domnului profesor Octavian Popescu, membru al Academiei Române, care mi-a oferit indicații importante și mi-a verificat atât textul subtitrării cât și întrebările, dar și ipoteza prezentată în partea finală, urmând ca eu să suport consecințele acestui act de îndrăzneală. Rezultatul a fost unul neașteptat de promițător și a generat la rândul său alte consecințe foarte benefice pentru ceea ce ar putea constitui în viitorul apropiat un proiect de studiu important. În funcție de rezultatele pe care le va obține echipa de cercetare ce va aborda studiul acestei ipoteze, se va putea oferi un răspuns mult mai clar legat de vindecarea maladiilor degenerative, care momentan sunt considerate maladii incurabile. Există speranțe. Știința face pași uimitori în ultima perioadă, iar așteptările rezultatelor nu vor mai dura zeci de ani, cum a fost în multe alte cazuri. Fiți alături de noi data viitoare la o nouă întâlnire cu Știință și Cunoaștere.